right, being recorded. The things that we really want to kind of hammer out today, as well as, again, big picture things. All right, so I'm going to go through. I'm actually going to delete this slide because that means nothing to you guys right now. I'm going to insert a new one in there. And whoops, not that way. Insert new slide, blank one. That way we can utilize that bad boy today if we need to. Okay, so the big question come up that we were going to discuss, again, why genes, how genes basically turn on and how they turn off. Okay, and there's a bunch of different models. You guys went through 3 models yesterday of how these operations, or what they call operons, primarily operons basically referring to more of the bacterial, like prokaryotic organisms, but basically how they work, okay? So that's going to kind of be our job today is we're going to break down, again, the lac operon versus the TRP operon. We talk about how that compares to eukaryotic organisms, just the many ways or many things that can happen to the eukaryotic. And that's probably the toughest thing about the eukaryotic organism is that we can't really give you like a definite answer to how it works because there is so many models and there's a lot of things we just don't know. So when we start looking at it, we try to break this down as far as what the bacteria is doing because of the simplicity, basically of their DNA, the simplicity of just how they work, right? And that's kind of where we are, where we're going to roll with today. So, but a couple things, let's go this way. A couple things we want to make sure we are getting today is I think we need to talk first and foremost, we're going to start out the differences between the prokaryotic and eukaryotic organisms, okay? As far as how they function, how they work. But you also got to go back to, kind of the beginning as far as like, well, what's the differences behind, right, the whole prokaryotic versus eukaryotic organisms. Because you remember, when we start talking about gene regulation, okay, we're talking regulation of the gene, right? So we're talking about any type of manipulation, any type of basically turning it off and turning it on. And that can happen basically all the way back on the DNA molecule. And we'll talk about that today, how there can be modification to the chromosome or chromatin, right? That can alter, right? We can take this all the way back to transcription, right, to where we know that there's RNA splicing. And why is there RNA splicing?
We can take out the introns.
Take out the introns, good. And then we also know there's these additions of the methyl caps, right, or the poly A tails that are, that's gene regulation itself, right, because you're manipulating, right, the strand, okay? And again, it's going to aid, if they don't have that methyl cap, more than likely the ribosome is not going to attach to the RNA molecule, and then that process will happen. Okay, when we get to translation, we start to see as an end result mRNA degradation. We start to see the protein degradation that when it's all done, right, they're going to be degraded to make sure that it doesn't just completely keep rebuilding because we've learned, and we learned a little bit yesterday, we'll learn more in the future, that things are good in our body in moderation. That if they actually increase in numbers, they can become unhealthy, become toxic, right, in some cases, in some situations. So again, we've got kind of this little model going on, right, that we're going to see, right, amongst, the whole, kind of as far as where genes can be regulated. But again, we can also start looking at prokaryotic versus eukaryotic organisms and be like, well, one, this probably doesn't need to happen all that much in a prokaryotic organism, okay, where we need to modify the chromatin. Okay, now you also got to remember what is chromatin and where does that apply? One, okay, we start looking at the reason why this wouldn't apply to prokaryotic organisms because they don't really have a very highly condensed, DNA molecule. It's relatively strung out in a single loop and so on like that. Enzymes can, or polymerases, enzymes can get to it and they can break things, they can build things up, okay, pretty easily with it. Okay, when we start looking at RNA, is there going to be any splicing in a prokaryotic organism? No, because what don't they have? They don't have any introns. They don't have any junk DNA, right? There doesn't need to be necessarily the caps, right? Because there's no nucleus. Therefore, anything that we have in transcription basically is going right to translation right at the spot. And so again, therefore, we're not really seeing much, in the case of RNA modification to get there, okay? And again, we're probably going to see some of these being relatively similar, right? So on down the line. So when we kind of start looking at it, ultimately, what we're probably going to look at, the one thing that is similar between prokaryotic and eukaryotic organisms is all about the process of being able to either turn the gene on and turn it off, right? And we start looking at the factors that can maybe manipulate that turning of the gene on and off. And that's kind of where we're going to go with a little bit today. All right. So here's where we're at. So we're going to break down prokaryotic forms of operons. Okay. And we're going to break this down into a couple of different things. Okay. So we can either talk about these operon models, okay, being, where's my little, is this it right here? Yeah, there we go. We can either talk about them being a positive regulatory process. Whoops, I don't know why I put position. Or we could talk about them being a negative, okay, regulatory process. And I'm talking regulatory because we're talking about basically, again, the process of regulating a gene. Now, when I think of positive and negative, right, I always go back to like positive versus negative feedback loops. In the case of a positive feedback loop, what's happening to, the overall end goal, or what's happening to the product? Yeah, keeping it going. You're enhancing it, right? You're actually building it up, okay? Or it's functioning, it's working, okay? If we're talking negative, what is negative referring to? Yeah, basically, we're going to try to reverse that process or stop, right, what is happening, right? So again, we talk about labor, right? Contractions increase. That would be a positive feedback loop because we want them to keep increasing, not we, but females want them to keep increasing because that's what ultimately is giving birth, right? Or helping them to give birth, right? In the case of, our temperatures, right? We want a negative feedback loop because as they're going up, we want to be able to stop that process and bring it back down to a normal rate because we know that obviously proteins become denatured at a certain temperature, body isn't going to function like it should. So when we start looking at these regulatory processes, again, positive and negative basically come back in the same type of approach. And so what I'm looking at here, though, is I'm looking at whether or not, right, that gene is turned on, right, or whether it's turned off, right, and how is it turned on or how is it turned off. So here's what we're looking at. Negative. Okay, gene is turned off. Okay, but it's turned off by, we're going to learn about this term. It's called a repressor protein. Okay, you guys learned about it yesterday. You should have been, or should at least know a little bit about it. Okay, and that repressor protein is actually activated. So a negative process, a negative regulatory factor, right, is where the gene is turned off by an activated, I'm going to put down, I can't use repressor protein because it's not the case for all. I'm going to put down by an activated regulatory protein. Okay? So the protein that regulates it. If I'm talking positive, that gene is now turned not off, but on. Okay? Gene is on. And it's the same thing, and it's activated, by N, activated regulatory protein. Okay? So now the question comes up, well, how is that regulatory protein activated? Okay, and that's kind of the ultimate goal that we're looking at is when we talk TRP operons, we talk LAC operons. Okay, what does it mean for that certain regulatory protein to be activated? Okay, now I want you guys to get your books out. Okay, we're going to look at this because your Pogos, if you have your Pogos, that's fine. You guys can utilize that as well because I don't have a very good chart. I don't have a chart up on the book for this one. So let's go to page 3. For this one, TRP operon 353 would be probably our best place to take a look. All right, we're going to talk TRP operon. We're going to talk LAC operons in this case. Okay. Now, I said there's multiple ways that we could basically kind of, we could list. We could, what's the word I'm looking for? Not constitute, not classify. Right. These regulatory, basically these processes. And I said there's either positive or negative. But then we've also got where it could be a repressor or it could be an inducer. Okay? And if something represses, what does that mean? It's going to stop, right? Okay, or it's going to slow down. Or we have an inducer. An inducer is going to do what? Yeah, it's going to increase or it's going to start, something along those lines. Okay. So we go through positive versus negative, right? Repressor versus inducer. Okay. We can have negative, all right. In this case, a negative regulation that can be repressor, or it can also be an inducer. Just depends on, again, what the overall goal is. And remember, both of these are happening primarily in, right, bacteria or prokaryotic organisms. Okay. So TRP, right, is a form of an amino acid. Okay, tryptophan, right, is what TRP stands for. Or TRP stands for tryptophan. Okay, which, the old myth, I would say it's a myth. I mean, it's pretty true. At Thanksgiving, you guys eat turkey, right? When you eat turkey, you generally get really, really tired and you start to take a little nap and you get sleepy-eyed, right? Well, what does that do to you? Nobody's ever heard that before? Oh, it's usually due to the tryptophan. Yeah, that's found in the turkey, right? So again, turkey primarily meat, right? Protein, right? It's an amino acid that you find inside of there, and that's usually what leads to the sleepiness inside of, basically your body when we're going through this. Now, here's what's happening in the TRP operon that we find in E. coli. Okay, so E. coli again is bacteria. We find E. coli in our body. There's good E. coli. There's bad E. coli. Right? We got good E. coli that we're going to work with our PGLO gene here, hopefully at the end of the week. Right? There's bad E. coli that can make people very, very, very sick. It just depends on what strain it is. But here's what's happening. In the case of tryptophan, okay, it is a repressible operon. Okay, so think about it. It's repressor. Okay, so let's break down what that means. So there's a lot of things going on here. Okay, one, there's a promoter, okay, which we've talked about in the past. The promoter is basically going to do what? It's part of a DNA sequence, and it's there. I'm looking right on page 353. I'm looking at basically where it labels promoter, right? And it's going to help simply to start the process, right? I mean, that's kind of what it's going to do, right? It's kind of the gene or the sequence where this gene is going to start being conducted or basically turned on, right? Or, simply transcribed. Let's go that route, okay? Now, so the ultimate goal is that if the gene is turned on, mRNA is conducted. That eventually goes on to form a protein, right? Okay. If that gene is turned off, generally it won't be turned into mRNA and so on down the line. Okay. So here's what we have. So in the first box up there, okay, we are looking at a couple of things. We're going to look at the promoter, okay, which is actually kind of a couple of different parts. Right? In that part, you're going to see that there is obviously the RNA polymerase. And then there's also this little area that's called an operator. Okay? Now, if the operator, okay, in this case, right, has nothing attached to it, right, that RNA polymerase can now work itself down that DNA strand and it can form an mRNA molecule, which will eventually go on and form the protein. Okay. Now, the question is, well, what would be bond to the operator? Okay. When we go down and take a look, there's this protein, right, that's in the red, and at this point in time is inactivated. Okay. And how do we know that it's inactivated? One, because it's not in the shape, right, that could fit into that operator. Do you guys see that? How it kind of is skewed a little bit, that it wouldn't fit into that operator. So therefore, it's inactive. Okay. So the question should come up, well, what activates it? Okay. And if that molecule is activated, what happens to the overall production of the mRNA sequence? Okay. So let's jump down. Okay. Now, when we take a look at that, we've got in the purple box, you got a promoter, right? Promoter sequence. We've said in the past, there's like the TATA box, right? TATA, that could be a promoter sequence, right? That's where the RNA polymerase knows where to attach to, right? Okay. Now, in this case, what do we start seeing happening? Okay. If you look down the line, the RNA polymerase is there, but something is now blocking it. Okay. And what's blocking it? The what? The repressor protein, that protein. Okay, which has now taken on a different shape because now there is a molecule attached to it that has now changed its shape to make it become activated. Okay, and what is that molecule that formed it to become, or basically made it now become activated? Which is, in this case, it's TRP, it's tryptophan. Okay, so in the absence of tryptophan, that protein is inactivated, but the gene is turned on. If there's a high amount of TRP, that means that regulatory gene is now activated, right? It's repressed. That goes up to that little operator. It's going to block the RNA polymerase, basically meaning that the gene is turned off. So let's think about the big picture. Okay, why would the gene be turned off when there is a large amount of TRP, and why it would be turned on when there is a small amount of TRP or hardly any whatsoever? Okay, good. If there's not enough TRP in our body, we can do what? We can make it. We can build it up, and we can basically have it there to be used to build other proteins. Exactly. And when there's enough, why expend energy on something that we don't need to build if it's already there? So in the case you eat turkey, large amount of tryptophan, guess what? That gene more than likely is going to be turned off because you don't need to make it anymore. Okay? So this is a negative regulatory process because in its activated form, what is that gene? Is that gene turned off or is that gene turned on? It is turned off, right? In its activated form. But it's a repressor molecule, or repressor model, because what happens? The TRP actually represses or it turns that gene off. Does that make sense? Yes? Okay. So there's a TRP model. Okay? And it's a repressor, like we just said, all right? It's going to meant to basically, if there's enough of it, it's going to have a negative effect on the production, on the breaking down, the building up of the TRP. Okay, let's jump. Okay, let's go to lac operon model. Okay, still finding in E. coli, we still find in a bacteria, relatively simple process here. So in the case of the lactose, okay, if we take a look up top, it is still a negative process. Okay, so which means that when that molecule, when that repressor is activated, or when it's active, right, that gene is still turned off. But now what we're looking at is not a repressible molecule or model. Now we're looking at an inducer, meaning that if there is a high amount of lactose, it's going to turn that gene on. Okay, and here's the tricky part about it. Okay, because again, there's a lot. I mean, this is about as complex as it gets, right? In my mind, as far as this class goes. So in the lac operon, here's what's happening. I'm going to jump. Let's go to A. So we don't have any lactose. Okay. Notice that the repressor molecule, in this case, or the protein, right, is in its activated form. So now it can bond to the operator, right, and RNA polymerase can't do its job, which means that the gene is turned off. Okay, active form, gene is turned off, negative model, right, negative process. Okay, but here's the catch. A little bit different, right, than what we've seen with TRP. Because remember, if there's a high amount of TRP, okay, that activated it. But here, if there is a large amount of lactose, it inactivates that protein. And if it inactivates that protein, right, what are we seeing? It can't bond to that operator, which means that RNA polymerase can't go through, which means that that gene is turned, in this case, turned on. If there is hardly any lactose whatsoever, that activated form attaches to the operator, RNA polymerase can't get through, and that gene is turned off. So now let's think about that process. Okay, if there is a large amount of lactose, why would we want that gene to be turned on? So we can break it down, because we're then going to build the proteins or the enzymes to break down lactose. And why would we want to break down lactose?
Make it more usable.
What would its use be? Nope. OSE at the end. Sugar. Yeah. So if it's a sugar, what's it used for? It's used for energy. Exactly. Okay. So and again, when we're looking at this, right, if there's enough of it, right, we want to keep that gene turned on because we want to build the proteins, the enzymes to break it down. But if there are hardly any in there, we're not going to continuously build those proteins and break it down because there's nothing to break down. Exactly. Yes, ma'am. Yeah, that question came up today. So when you talk about lactose intolerant, remember there's a couple things going on here. And somebody's like, well, what does that mean? Or what happens? Well, there's a lot of things that you can kind of dictate with being lactose intolerant. But basically, you are not tolerant to lactose. Okay, which means that your body can't break it down. And if we're going to bring this back into, the genetic, reasoning behind that, more than likely it's because that gene has been manipulated. So you can't build the proteins to break down that lactose. Okay, which means that what do? If you have dairy products and you're lactose intolerant,
Get an upset tummy.
yeah, you get upset tummy. Okay, diarrhea, right? Probably fart a little bit, right? Probably pass some gas, little things like that. But here's the other catch. I mean, are you out of the water if you can't break down lactose? No. Because what's the major molecule that we do break down?
Okay.
We break down glucose, okay, which is actually going to spin us into the next little deal here, okay? So it's like, okay, so you don't have lactose, we still break down glucose primarily anyway. So now I guess the question comes up is, well, if we already break down glucose, why do we need to break down lactose?
That's what we get from our mothers.
Sure.
Gives you strong bones.
Yeah. Okay.
Because cheese is really
Oh, okay. There we go. Okay. Yeah. No, but why would we? Why would you? If we already break down glucose, when in this situation would we have to break down lactose?
Because they don't get enough vitamin C because they don't get enough sun.
No, not necessarily that. Nope.
Breastfeeding.
Nope. I mean, obviously, if we take in lactose, we're eventually going to have to break it down. There's no doubt about that, okay? But the point I'm getting to is that if we primarily break down glucose first, when would we start to break down lactose? When we're out of glucose. Exactly. Okay? And it's situated like we're out of it. So we wouldn't necessarily utilize this until we have to. Okay. Now, that's where we start to tie in, right, this positive, right, model or this positive type of process. Because every single one of these up to this point were considered negative, meaning that when that molecule and that protein is in its inactivated form, it turns that gene off. Okay? But lactose works a little bit differently because in order for the lac operon model to actually function and work, that gene has to be turned on by something else. And you guys have already answered that question. What was going to turn that gene on, or what's going to allow that operon, that model, to function? If there is not any glucose in our body. Okay, so we're going to jump a little bit here. Okay, well, let's go to page 355. Okay, and 355, now we start looking at this model, okay, of the lac operon. Okay, so a couple things. Okay, one, I mean, in order for the lac operon model to be turned on anyway, we know that we need a, whoops, I don't know what, just let's get out of there. We know that we need, I didn't want to go there. Okay, we know that we need a high amount of lactose, right? Okay, and in this case, we also need a low amount of glucose. If there's a low amount of lactose, this model, it doesn't matter what happens, it's not going to be turned on because the lactose isn't there to inactivate that protein, which will eventually keep from going on, right? Okay, now, here's what we're looking at, though. So there is basically another basically in this case, another molecule that is going to help this process basically turn on. And it has to happen first, along with the high amount of lactose, and obviously that's a low amount of glucose, okay? So a couple more molecules that come up, okay? There's what they call the cyclic AMP, CAMP, C-A-M-P, right? That's going to come into play. And then you've got what they call just the CAP, which would be basically an acceptor protein. Okay, so it's like the CAMP acceptor protein. So if we take a look, the little blue kind of oval there, right, would be what we call the CAP. It's an acceptor protein that accepts, all right, cyclic AMP. All right, well, now we've got to ask the question, well, where does cyclic AMP come from? Okay, so when we start looking at basically the cyclic AMP, right, and just go through and just read real quickly on this, all right, how does E. coli cell sense the glucose concentration and relay this information to the genome? Again, the mechanism depends on the interaction of an allosteric regulatory protein. Big word there. What does allosteric mean? Protein does what?
What paragraph is it?
I'm looking at right underneath positive gene regulation. It's the second paragraph. Well, we've talked about allosteric before. And what does allosteric mean? We talked about it with enzymes. Because this is going to come up a lot here in the next month and a half. So if we're looking at an allosteric like mechanism or an allosteric protein, it is one that is non-competitive and it allows that enzyme to change shape. Okay? So if a molecule attaches to it, it's going to change shape. If a molecule doesn't attach to it, it's going to go back to what I would consider to be its normal shape. Molecule attaches, it changes shape. Okay, so it's an enzyme. It's a protein. Okay, now, so in this case, the cyclic AMP, which accumulates when glucose is scarce. Okay, so when there's not a lot of glucose, the cyclic AMP, the CAMP, right, actually increases. And when it increases, what does it do to the cap? It attaches to it, exactly. And when it attaches to it, what does it do? Does it activate it or does it inactivate it? It activates it. Okay, it changes shape and it activates it. Okay, now when we take a look, when that molecule is activated, right, the RNA polymerase can attach to it, all right? But until then, that RNA polymerase will not attach. So if there is, again, a high amount of glucose, that means that there is a low amount of CAMP or this cyclic AMP. That also means that it's not going to attach to that cap. That cap is going to be inactivated. RNA polymerase is not going to attach to it. So it doesn't matter how much lactose is in the system, it's not going to work. Okay? In order for the lac operon model to actually continue on and function, there has to be a low amount of glucose, which means that there's a high amount of CAMP, let's just say, which is going to attach to that cap, which is an acceptor protein, which is going to bind to the operator. Now not to the operator, but the promoter, which now allows the RNA polymerase to attach to it. And now if there is enough lactose, that is now going to, be broken down. Okay, and it's basically going to, again, do what we just seen, right, when we talk about the whole lac operon model. Okay, so confusing as all get out? Yeah, sure, absolutely. Okay, and if I'm you, I'm probably going to read up on this because guess what I have to do every year when it comes to this? I've got to read. I got to go about an hour into the book, and I got to go back and make sure I've got things straight because it's as confusing to me every single year when I teach this because there are so many, like, just ins and outs in the way that it works. Okay, yes, ma'am.
So, will lac operon be positive and negative?
Absolutely. Yep. So positive in the form of when glucose is low, all right, that basically forms the CAMP, right, which binds to the cap. That activates that allows the RNA polymerase. Now, so that part's positive, okay, because it's going to activate it. It's going to basically say, hey, that gene is turned on. But there also has to be a high amount of lactose, right? And if there's that high amount of lactose, what's it going to do? It's going to inactivate that repressor, which basically means that gene is continuously turned on and it's going to break that baby down. If there's not enough lactose, that molecule, that repressor stays active, right? And it's going to shut that gene down, which is where we get into the whole negative side of it. Okay, so the low glucose will activate it. The high lactose will keep it turned on. Yep. Okay. Yes, ma'am.
What happens when you run out of lactose and glucose?
Well, what does happen? You tell me.
Break down fat.
Or is there just nothing?
No, because we have reserves, right? First of all, we got that 24-hour supply that we find in our liver. Right, we've got why can't I think of it right now? We've got fat that eventually we end up breaking down as energy reserves. No, I mean we have things that are there for us.
Protein.
Yeah. Say it again.
What happens when it's just all gone?
Well,
I think that's pretty. I think that's I mean, what happens when anything? All of it's gone. You die. You have no energy. You have no energy. You just die. Yeah, I mean, simple as that. So. And again, which is why you constantly have to take in food. And again, if you are on some maybe like keto diets and stuff like that, as far as your, the diet fads, what are they doing? Right? You cut out carbohydrates, right? You're basically just taking on straight fat. Why? Because once you start to break down the carbohydrates, okay, you then, once you stop breaking down carbohydrates, your body then resorts to the fat. Exactly. Okay. So that's kind of where we are at. Oh. Okay. All right. So now the question comes up. Okay. So there's your prokaryotic. Okay. Now, what do we know about eukaryotic organisms? Okay. And ultimately, where do they come into play? Okay. Now, the models that we start looking at are not as simple as we've seen with the whole lac operon and the TR operon models. Okay. What we start now focusing in on is that we've got a lot of things going on in the whole kind of eukaryotic system. And I think it makes sense. Okay, we're much more complex organisms, right? So therefore, everything about us is going to be much more complex. And we can start looking at the whole gene regulation, right? We could take this back to the big picture that ultimately, right, genes are going to be turned on in certain cells, right? Well, what basically is going to dictate what genes are turned on in certain cells? A lot of it is the environment that those cells are put in. Okay, we can go back to the question of, well, if you've got two different cells that have the exact same form of DNA, right, how do they have different characteristics? Because certain genes are turned on in one, where certain genes are turned off in the other. So now it's like, well, how does it regulate what genes are turned on and what genes are turned off, right, in each of those cells? And again, a lot of that has to do with the environment that it's found in. Okay, and so when we start looking now at the eukaryotic models, right, there is a lot of different ways, again, a lot of different parts in which genes can be regulated. And ultimately, when I made this little chart right here, I was looking at the slide, or I was looking at the picture on page 356, right? And all the ways, again, that modifications can occur that can ultimately manipulate that of the gene, all right? Simply turning it on or turning it off. But I think the first thing that we need to understand is that when a gene is turned on, that's happening at the level of the DNA, right? And so what's going to cause genes to be turned on or simply genes to be turned off? We got hormones, right? We know that there's chemicals that are produced inside the body that'll make that happen. What else? Yeah, and we'll get to that here in a little bit. Okay, and that's kind of a part that I want to talk about. But we've got environmental factors, right, that can turn genes on and turn genes off. Diets right now can turn genes on, turn genes off. One of the biggest research that's coming out right now, obviously, is the whole COVID situation, but it's not about the sicknesses. It's about what's happening to people because of the wearing of masks, right? The dermatitis, the whole dermal kind of problems that's going to come up. You've got basically lesion sores. You've got acne that can build up. You've got, there's a constant washing of your hands and using hand sanitizer, which is causing dry skin and all these other things going on. And so now you start looking at, well, what possible causes or what possible problems could that lead to in the future? And again, they're turning on and off of genes possibly in the future. You start looking at diets, right? Taking on certain foods. In this case, obviously, if you can't take in lactose, right? We know that operon can't ever be turned on probably, right? Because again, you don't have the ability to break it down. And so there's diets, there's health, there's exercise, there's, again, COVID, there's environmental formulas that can basically, again, right, cause certain genes to be turned on and turned off, right? That we generally call epigenetics. Okay? And so if I have the term epigenetics, and it's on your guys' slide, it's on your guys' notes, that basically is referring to any form of gene manipulation where it doesn't change the DNA. Does that make sense? It can turn a gene on and off, but it's not going to manipulate. And so it's different from a mutation. Right? In terms of when we start looking at mutations happen, and when you go on, right, and depending on where that mutation occurs, it could ultimately get passed down from generation to generation to generation, right? Just depends on, again, if it happens in the reproductive cells during production of the gametes. When we start looking at this whole epigenetic process, it's not going to be passed down because it's just more about turning genes on and turning genes off. So it's probably more of like an adaptation than it would be, or an acclimation in that case, that versus something that you're passing down, genetically from parent to offspring and so on down the line. And any of those basically will come up, right? And again, I provided you guys with a nice little website down there if you want to, right, take a little bit more look at this, because I think it's kind of phenomenal. I think they've got a lot of good research, a lot of things that can kind of talk about what can turn those genes on and off. If you click on that link, it's going to give you the diets. It's going to talk about environmental stimuli. It's going to talk about, toxins, and so on down the line that can ultimately have that kind of a change. Okay, aging, drugs, you name it. Okay, now here's the kind of the in-depth stuff, okay, that we're going to look at. So if we're going to talk about primarily a gene being turned on in a eukaryotic organism, okay, there are a lot of different, again, things that are involved in this process. Okay, that you guys went through. You guys listened to Paul Anderson yesterday. Let's just make sure that we have these parts down. Okay, one. There's going to be an enhancer. There's going to be a promoter. And then there is basically the DNA sequence. Okay? You guys with me on that? So none of this is going to occur, okay, unless this enhancer and these promoters come in contact with each other. Okay? And as we talk about this whole junk DNA. Well, now they're starting to, again, figure out, more research is being done. That so-called junk DNA is serving purposes. It's non-coding DNA, but it's still DNA sequences, right, that basically will activate a gene. It's going to turn a gene on. It's going to help in some way, shape, or form turn a gene on, okay? So we've talked about promoters, for example, the whole TATA box, right, TATA. That's needed. That's what an RNA polymerase is looking for in order to come in and in order to build up that molecule. But in order for that to be activated, in order for that promoter, in order for that RNA polymerase to attach, it's not just looking for the promoter. Something else has to happen. That enhancer has to bind to that promoter. But the problem is that enhancer could be hundreds of base pairs away from the promoter. So again, how does it activate it? Okay, well, first and foremost, we've got these little molecules called transcription factors, or regulatory factors is what they could be called. Regulatory referring to the regulation of the gene, transcription factors referring to that they're going to start activating that DNA molecule, which if they activate the DNA molecule, then they're going to be able to go through the process of transcription, which will eventually lead to translation, which will eventually lead to, again, the building of the protein itself. Okay, so I think what's also kind of unique is that people are asking, where do these transcription factors come from?
They were always there.
I mean, they're there, okay? And they're different, right? Because, again, they're different for each gene. They're different for each organism, but they're also different for each gene. Okay? And they're proteins, which means that they have to be made by what? Ribosomes. Which, how do the ribosomes know what proteins to make?
RNA.
Because of the RNA. Where's that RNA come from? It comes from DNA. Well, how does DNA know which ones to make? Whether that gene is turned on or off. Yeah. So when we start breaking this down, we actually can take this all the way back, not just to the transcription factors, but now we've got what they call regulatory genes. All right? And the regulatory genes are genes that are going to make what? The transcription factors, right? And so again, how does it know? Yeah, I mean, one, you start looking at the environment it's found in, right? When you start looking at, again, you've got one cell that turns into two cells, and it's going to basically, again, it's going to kind of transfer that down, right? And so the environment that it's living in, basically, its climate. Remember, every cell has kind of its own internal little climate, right? Its own little system. And a lot of that's built up, again, just based on what genes are turned on and off. And we'll talk about cell specialization a little bit more, again, when we get to cell communication here in the next couple of units. But, I mean, it's obviously something to think about. So those transcription factors won't actually they're actually built by a regulatory gene that's, again, found on the DNA. So once they're built, and again, you guys can see this. You can look at the same thing Paul Anderson basically showed yesterday, right? Is that what's going to happen in this case, and you look at page 360, is that once those transcription factors come in combination or come in contact with that enhancer and that promoter, they're basically there to kind of almost take that DNA and allow it to bend back on itself. All right. And once that enhancer comes in contact with promoter, now we can see that RNA polymerase is going to be attached. Right. And now, right, we can see, right, to where it's going to go on, it's going to transcribe that molecule. But again, that's what's needed. All right. It needs those type of transcription factors. Right. The one example they give you in the book, I think it's kind of neat to, just even thinking about, we take a look at a liver cell, take a look at a lens cell. Right. You've got two different genes here. One makes albumin, right, for the liver. Right. The other one makes crystalline, right, which is another molecule. Again, how does it know what to make? Well, it's all based on the transcription factors that are produced, right? There's transcription factors, right, that are specifically there to combine that enhancer to the promoter of the gene that is going to produce that specific molecule, right? A liver cell isn't going to have the transcription factors that the lens cell has. The lens cell isn't going to have the same transcription factors that the liver cell has, so which means that it can only turn certain genes on. And again, what dictates that? The regulatory genes that are producing those. And so again, we'll take that further and further as time goes on, right? And kind of talking more about how those cells become specialized when you've got just the one cell, which turns into two, which turns into four, and so on down the line. How do they become specialized, and how do they know which transcription factors to take on? Okay? Questions? Oh, perfect timing. All right? Yeah, I think we're good. I think we're done there. Now, again, a lot that we kind of threw at you guys today. But again, this is, I would say, probably some of the more complex days all right, or complex, information that we are going to go over. Okay.